So in our learning unit on amino acid metabolism or protein metabolism, we're going to take a very similar approach to what we did with lipid metabolism. We're going to first consider sort of exogenous and endogenous sources of protein. And then once we get to amino acids that are inside cells, just like when we got to fatty acids inside cells, what happens with those is irrespective of where the original source was. So whether it came from exogenous or endogenous sources. So again, we're going to focus on the process of amino acid deamination, because that is going to allow us to break an amino acid into its two sort of component parts: nitrogen, which is disposed of in the urea cycle, and then the alpha carbon skeleton, which is broken down or used for other biosynthetic processes in other ways. We'll close out just kind of like talking about other pathways that maybe involve nitrogen, and then specifically the concept of nitrogen fixation. So we begin this learning unit after we discuss sort of our fundamentals pieces, just doing another comparison of carbohydrates, fats, and protein. And again, looking at proteins, now this kind of rounds out the three main macromolecules that we kind of see in our diet. So even though we did a good bit kind of comparing and contrasting glucose and fatty acids and triglycerides in our last chapter, we kind of bring in amino acids in this chapter, reminding ourselves that from a caloric standpoint, they're worth the same as carbohydrates. But again, remembering that the purpose of consuming amino acid or consuming proteins is not necessarily that they're used as a fuel, but really that they are used as a building block. Not only a building block for other proteins, but a building block for lots of other important biochemical metabolites, which may be amino acid derived. So an important concept to think about here is, as much as we have intended storages for carbohydrates and then lipids in our body, we really don't have intentional reserves of amino acids and protein in our body. We do have what we were going to refer to as unintended reserves. That is, if we do need to tap into our own sort of tissues in order to access amino acids, we actually are going to see selective atrophy of liver and kidney as tissues to provide amino acids as needed. And again, when we talk about what these might be needed for, it might be that we need them either for energy or to make them into glucose via gluconeogenesis, or you actually might need to tap into them because you just need those building blocks to make new proteins or make new amino acid derivatives. And so, again, not a lot that I'm going to go through on this slide because we did a pretty deep dive on the comparison of glucose and triglycerides in our last chapter. So just kind of building on amino acids here. So like we had in our lipid metabolism chapter, I like to think about sort of exogenous versus endogenous sources of protein. And then once we break down those proteins, breaking those peptide bonds to give us free amino acids, once we have those intracellular amino acids, then what we do with them is really the same regardless of the source. And so again, not a lot that we're going to discuss with exogenous sources or dietary sources of protein. We do need to think about their digestion, their uptake, and their transport, but we don't have as many issues with hydrophilic species like amino acids like we did when we had triglycerides. We will spend a good bit more time talking about sort of the endogenous pathway. So whether it's enzymes or tissues that need to be turned over, we do see some degradation of processes in this regard. And so there was a homework question that asked us to think about, well, when might that happen? Why might that be an important process to sort of have the breakdown of internal proteins? And so three things that I want you to think about for that are, it is a mechanism for eliminating proteins that maybe are malfunctioning. So maybe if they've somehow become damaged by environment or something, we can actually degrade them. It's also a way to regulate metabolic pathways. We've done a lot to understand how enzymes in metabolic pathways are regulated by activity. So phospho-dephospho states, thinking about R state, T state, and so forth. But remember, long term, we sort of talked about another mechanism for regulating metabolic processes is to regulate the availability of enzymes. And so not only is part of regulating metabolic availability, upregulating the transcription of genes to make new proteins, but it's also upregulating the degradative processes that will get rid of those genes or get rid of those proteins. And something that we'll spend a little bit more time talking about the details of in our last learning unit, sort of when we talk about some applications of integrative metabolism, is we'll talk about, well, if you are in a situation where you're fasting, potentially utilizing endogenous proteins are a way to tap into a reserve when that's needed. So one of the substrate cycles I want us to sort of think about here is this concept that we can have proteins that are, again, either exogenous or endogenous here, and we can have these proteins that are catabolized to make amino acids, and then they may be synthesized into other proteins or maybe other amino acid derived metabolites. But the thought is that we want to sort of maintain a fairly closed loop here. We don't necessarily want to have a lot of amino acids that we need to degrade. Ideally, we'd have sort of a perfect cycle of consuming just what we needed in terms of proteins, or taking and degrading just what we needed in terms of our own endogenous protein sources, and really not have to have too much excess amino acid that we need to worry about getting rid of. Nonetheless, if we do need to break these down, they do have, again, a caloric value of 4 calories per gram. But when we look at the structure of an amino acid, we see that we have what we're going to refer to as an elemental problem. So again, when we think about carbohydrates, we think about carbon, hydrogen, oxygen. When we think about lipids, we think about pretty much just carbon and hydrogen. So when we add in our amino acids here, we have carbon, hydrogen, oxygen, and then nitrogen. And so that nitrogen really kind of introduces a new element that we really have to think about how we're going to metabolize. And so when we think about sort of just the general structure here of an amino acid, what we really have is the alpha amino group that we need to think about disposing. And once we take care of that alpha amino group, then we've got this alpha carbon skeleton. And this carbohydrate portion really sort of serves its secondary function based on who it looks like. And so we're going to kind of spend a little bit more time in a later learning unit talking about this concept of glucogenic versus ketogenic amino acids. And basically, if you are an amino acid that can be converted into pyruvate, that means you can ultimately be made into glucose. So that's hence glucose and glucogenic amino acids. So again, remembering that pyruvate can be made into glucose via gluconeogenesis. If you are an amino acid that can only be made into acetyl-CoA, then you are referred to as a ketogenic amino acid, remembering that acetyl-CoA can be made into ketone bodies. And so that's where the ketogenic part comes from. So again, these are amino acids that can be made into different hydrophilic circulating fuels, but again, some of them being glucogenic can be made into glucose. Other ones that are ketogenic can only be made into ketone bodies. And again, we spent lots of time talking about the different local versus global fates for acetyl-CoA that's produced in a need state. Again, using it locally, being broken down via the citric acid cycle and then electron transport oxidative phosphorylation will allow for the production of ATP locally. Or you can have ketone bodies that are synthesized in the liver that are put out into the circulatory system for global use by any tissue to be able to utilize and make their own energy. And again, if there's excess, we still can store that acetyl-CoA as triglycerides. So again, this elemental problem, the last little thing to just highlight in this slide, we will get into the details of in a little bit. But when we deal with this elemental problem, we basically are going to dispose of those nitrogen atoms as urea through its synthesis in the urea cycle. All right, so kind of a busy slide here, but just kind of going through and highlighting some important aspects here of exogenous versus endogenous sources of protein. So let's start over here sort of on the left side with our exogenous proteins and thinking about proteins that we might consume in our diet. So just like we walked through sort of what happens when you eat some lipids in your diet, we're going to walk through and kind of talk about the same process that happens with regards to proteins. So again, proteins within the stomach encounter the enzyme pepsin, which helps to break that peptide bond. And then we learned about our serine proteases that are synthesized by the pancreas and then secreted into our gastrointestinal tract, where they act in the small intestine to continue to break apart peptide bonds to give us those free amino acids, remembering that free amino acids are what are necessary in order to get across that one cell thick membrane in our gastrointestinal tract. So kind of maintaining that integrity is very important. So only monosaccharides, fatty acids, and amino acids are really able to get past that gastrointestinal one cell thick membrane. And this concept of leaky gut is really when you have species that are larger than that entering the bloodstream. And particularly this is important in this chapter because as peptides may be able to enter the bloodstream, they can cause significant inflammation and elicit an immune response. So again, that is part of the problem with this concept of leaky gut. All right, so a lot of that we've talked about already, so just kind of reviewing these pieces that have to do with sort of endogenous delivery of amino acids into our cells. But some new things that we haven't talked about are sort of endogenous sources of protein, enzyme, and amino acids within our body. So again, highlighted this already, but just put here on this same slide the three purposes, the three reasons that we might have for breaking down sort of internal proteins in our body. And again, whether those are proteins found in tissues or enzymes that we need to have undergoing turnover. So just a little bit of a distinction between non-specific sort of degradation pathways versus specific degradation pathways. So what I want you to remember are the two different organelles that are responsible for non-specific versus specific degradation. So non-specific degradation involves the lysosome. We saw the lysosome in our last learning unit on lipids when we talked about receptor-mediated endocytosis and we talked about the fusion of those particles with a lysosome to facilitate sort of the degradation of the particles within. So lysosome is often called the stomach of the cell. You know, we give these little nicknames to some of the organelles that are there. And again, remember the reason for that is because the lysosome contains a fairly high pH and it contains a lot of degradative enzymes. And so how things sort of get targeted to go into a lysosome sort of has to do with some of the things that might be going on in the body. So if you're in a time of need, and we're talking about that selective atrophy of liver and kidney tissues, the lysosome is partly responsible for sort of degrading some of those tissues to generate the amino acids that can help with that need state. We also have some time of plenty situations here. There's this tag, and you don't need to remember the name of this, but if you were to see it, recognize that this is a tag that might be on some proteins. Again, these represent the amino acids, some unique amino acids that might be there that would allow this protein to be tagged as a protein that would go to the lysosome for degradation. So just some interesting examples of lysosomal specific degradation. We talked briefly about uterine regression. So again, the uterus shortly after birth weighs about 2 kilograms, and within about a 9-day period, it's degraded to about a 50 kilogram sort of more normal size. And the degradation of this tissue internally is facilitated by lysosomal degradation of that tissue. Something else to sort of think about is rheumatoid arthritis is actually, it's an autoimmune disease, but really what happens in that autoimmune disease is you have extracellular excretion of lysosomal enzymes into joint spaces. And so that's not supposed to happen, but again, this is sort of an autoimmune response, a hyper-extensive sort of immune response in this situation, then again releases these lysosomal enzymes into joint spaces that causes degradation of joint tissue, and again is what causes that arthritic situation. So again, lysosome being non-specific degradation. Now it's a little bit confusing because there might be some specific parts that allow us to sort of facilitate that degradation, but it really is a fairly non-specific process that you have a high pH and some lysosomal enzymes that operate well under that high pH. So more specific degradation, what I want you to think about is the proteasome. We're not going to go through the details of how this large assembly works, but it's basically a protein degradation factory. We can think about it almost like a machine that's going to degrade proteins that are tagged with another specific molecule. So if proteins are tagged with a molecule called ubiquitin, This is referred to as a death tag. That means that these proteins are then tagged and trafficked to the proteasome for degradation within the proteasome. So the proteasome, again, is a very specific machine that degrades proteins that are tagged with this ubiquitin tag. And so some proteins that might be sort of more easily tagged for degradation with a ubiquitin tag are what we call pest proteins. So just like this KFERQ tag, pest again refers to those specific amino acids that we might have there. And also the identity of the amino acid on the N-terminus can have some influence as to how quickly a protein is turned over. And again, not anything that you need to know. You do not need to know these specific amino acids, but certain amino acids are going to give a protein a fairly short half-life versus some other amino acids indicate a fairly long half-life. Okay, so at the end of the day, remember why certain proteins inside our body might be sort of tagged or subject to degradation or turnover. And again, know the two different machines, if you will, organelles or machines that are responsible for nonspecific versus specific degradation within our cells. All right, so a very generic slide here, but again, a very important slide that kind of summarizes a lot of the important processes that are going to happen here. Remember, we've got 20 different amino acids, and what we're going to see here is we're not going to have machinery that operates on all 20 amino acids. What we're going to do is have some initial chemistry that allows us to funnel these 20 different amino acids down to two important amino acids, aspartic acid and glutamic acid. And that's going to happen via what we call transaminase chemistry. So we're going to look at that in the next couple of slides here. But that transaminase chemistry is essentially going to allow us to collect all of the amino groups as either aspartic acid or glutamic acid. And then we're left with the remainder of the carbon skeletons of those other 18 amino acids. And again, what happens to those depends on what they look like. Again, whether they're ketogenic amino acids, glucogenic amino acids, or maybe they're just recognized to be some type of a citric acid cycle intermediate that can be ultimately degraded to carbon dioxide and water and used for energy. As we mentioned before, once you do this transamination chemistry and you get down to aspartic acid and glutamic acid, these are going to be the two important amino acids that we use within the urea cycle to make urea. We're not going to go through any of the details of the metabolic enzymes within the urea cycle, but I do want you to know the structure of urea and how it is built from specifically aspartic acid, glutamic acid, and bicarbonate. So we'll see that on the next slide here. But again, highlighting the two important things that we're going to do with amino acids. And it kind of harkens back to this slide that we had on, or this image that we had on one of our first slides there. Thinking about this substrate cycle occurring between proteins being catabolized to their amino acids and then resynthesized either into other proteins or other amino acid derivatives. So again, that's that rebuilt component. Okay, so we might have some amino acids that we're going to rebuild into something else. Or we can think about recycling them. And so that's sort of this blue component here, where if we have amino acids that are in excess, well, we're basically just going to recycle them into fuel products. And so we're going to convert them into carbon dioxide and water and sort of get rid of that excess. All right, a busy slide here. So let me sort of break it apart again. We had a few homework questions sort of dealing with this. Remember on our last slide, we said that we're basically having 20 different amino acids, and we're going to funnel them to either glutamic acid or aspartic acid. I want to make sure to highlight here the differences between when I say the term glutamic acid and I say the term glutamate. They fundamentally are referring to the same amino acid, but when we say glutamate, we're referring to the deprotonated form. Glutamic acid refers to the protonated form. So I might, again, interchange these two terms, but just understand that really we are referring to the same amino acid. It just really has to do with the protonation state. All right, so transaminase chemistry, okay? What ultimately happens here is we're going to have two different alpha keto acids. So we're either going to have alpha ketoglutarate or oxaloacetate sort of come into play here. And what happens is we're going to take an alpha keto acid and then one of the 20 amino acids. And again, these alpha keto acids are either going to be alpha ketoglutarate or oxaloacetate. We do a transamination chemistry. Trans, you can kind of think about being swapping. We are swapping who has the nitrogen part of that amino acid. So again, we can see this a little bit clearer down here when we look at some structures. So here again is the structure of alpha-ketoglutarate, and here's the structure of oxaloacetate. Another way to think of these two guys is alpha-ketoglutarate is the alpha-keto acid, equivalent to glutamic acid, and oxaloacetate is the alpha-keto acid counterpart to aspartic acid. So at the end of the day, I want you to sort of see what happens here. This is our amino acid, right? We've got an alpha amino group. We've got a carboxylate group. Here's our alpha carbon with our alpha carbon's hydrogen, and then there's our R group. So at the end of the day, what happens is this alpha carbon, which was attached to this nitrogen, is swapped out for the alpha carbon that's now a carbonyl. So just look what we have here. If we've got these 5 carbon atoms, and again, we swap this guy out, not for a carbon nitrogen, but for a carbon oxygen bond, we get alpha ketoglutarate. So part of one of the skills in this chapter and why it's still important to know our amino acid structures is I do want you to recognize that, oh, this guy is the alpha keto acid skeleton of this amino acid. So alpha ketoglutarate is the alpha keto acid skeleton of glutamic acid, and oxaloacetate is the alpha keto acid of aspartic acid. All right, so again, what does this really mean here? Well, if we've got alpha ketoglutarate and oxaloacetate that are willing to come in and pick up the nitrogen component from any of the other 18 amino acids, what that means is they are going to become their component amino acid, either glutamate or aspartate, and then that incoming amino acid is basically going to be just left as its alpha keto acid skeleton. We'll talk about in the next couple of slides, well, what do you actually do with that? But at the start here, I just want you to understand the concept that we're going to have, and really, even though we've got that there are 20 amino acids, it's 18 of the other ones that are going to give up their amino group either to alpha ketoglutarate to become glutamate or to oxaloacetate to become aspartate. And that chemistry is called transaminase chemistry. So the enzymes are transaminases, and they require vitamin B6. So we learned vitamin B6 as PLP. This is why we need vitamin B6 to do this transaminase chemistry. Okay, so again, the rest of the chapter is going to kind of deal with, well, what do you do with these new alpha keto acid skeletons? They're carbohydrates, so what do we do with them? So again, we're going to address that question in a little bit here, but let's now talk about, oh, we've made our life easier. We've funneled 20 things down to 2. So now we just have 2 amino acids that we have to deal with. All right, let's first deal with glutamate because we have a little bit more work we need to do with glutamate. Glutamate undergoes oxidative deamination. So again, we're going to generate a reduced NADH for that. We generate an intermediate that I don't really need you to know much about what the intermediate is, but we have an intermediate that we're going to add water to, and we're ultimately going to get alpha ketoglutarate and then free NH4+, free ammonium ion. So again, glutamate undergoes oxidative deamination, and the products are NADH, alpha ketoglutarate, and then free ammonium ions. And this is important because this ammonium ion is going to be used to synthesize urea. Aspartate just gets used directly in the urea cycle, but the nitrogen in aspartate provides another nitrogen that's in urea. So again, here is the structure of urea. I do want you to know the structure of urea. A lot of what we're going to see in the next couple of chapters, we're going to call recipes for things. I want you to know the recipe for urea. I want you to know the structure of it, and I want you to know that one of the nitrogens comes from free ammonium that comes from the oxidative deamination of glutamic acid. The other nitrogen atom comes directly from aspartic acid that's going to be used in the urea cycle. And then the carbon that's found in urea comes from bicarbonate. We've seen this a good bit. Carbonate, which we have floating around pretty plentifully in our bloodstream, is going to be a great single carbon source. Okay, so in this case, urea cycle is going to use it to build urea. All right, so there was a lot on that slide. But again, if you need, go back and rewatch it, but hopefully that makes sense, that we've taken and funneled our amino acids to 2 that we're going to then use to make urea. So now let's talk about, well, what do we do with these new carbohydrates that we have? How do we handle taking care of those? There was just a couple of other homework questions that we just sort of handled a little bit like a laundry list here. I just want you to know sort of the names of different kinds of enzymes that you might have in your body, or proteins, regulatory versus housekeeping proteins or genes. So things that are regulatory are things that we turn on and off depending on what the cellular needs are. And again, we've learned that we can have short-term or long-term sort of regulatory control mechanisms. Again, controlling the activity via an on-off switch, or controlling the availability, as we just learned here, via increased degradation, or again, upregulating transcription and translation of genes. Housekeeping genes, on the other hand, are genes that are always going to be present, tend to always be on, performing sort of basic functions. So when people are looking at, for example, proteomics or transcriptomics, to look at what messenger RNAs or what proteins are present under a certain situation, housekeeping genes tend to be ones that you follow, that there should just be a baseline level of activity that's always there. And then you can look at, well, is this gene upregulated or downregulated in this certain situation? And then again, another homework question sort of just reminding us about the importance of maintaining 5 millimolar glucose, and then thinking about kind of glucogenic amino acids being the only ones that we can really use to make glucose and help with that situation. And again, that's because amino acids that are glucogenic can be made into pyruvate, which then via gluconeogenesis can be made into glucose, right? Both of these processes require some energy. So again, we can use endogenous triglycerides to help with that cost. All right. So again, how do we deal with the alpha keto acid skeletons that we're left with? And so another alpha keto acid sort of skeleton that I want you to recognize its amino acid kind of counterpart. Pyruvate is the alpha keto acid of alanine. So again, I want you to know that guy. You do not need to know the details of what's on this slide. You don't need to know which amino acids are glucogenic, which ones are ketogenic. Again, there are a few that we're going to talk about in some detail, but obviously the ones that I want you to know now are alpha ketoglutarate, again, which is related to glutamic acid, oxaloacetate, which is related to aspartic acid, and then pyruvate, which is related to alanine here. So again, we've got a lot of these that kind of live on the right side of the citric acid cycle here that are related to acetyl-CoA, and then ones that sort of live in the later part of the citric acid cycle are gluconeogenic. So, glucogenic. I should say you don't need to know which ones are which, but it is not, coincidental that these occur sort of in the first part of the citric acid cycle here. So just to highlight some stories that we are going to talk about, we are going to talk about metabolism of the amino acid phenylalanine as it relates to the metabolic disorder PKU. We are going to talk about glutamate a little bit more as we talk about how we synthesize amino acids, and we're going to talk about MSG for that. And then we're going to talk about methionine synthesis as that relates to MTHFR genes. So we talked about those last semester. All right, so again, thinking a little bit about the MTHFR situation. So some of this is information that we learned last semester, right? We learned about MTHFR being an important gene that's responsible for converting THF, which is tetrahydrofolate, into 5-methylfolate. That is the bioactive form of vitamin B9. So again, I'm not going to get into a lot of the details about why folic acid supplementation is problematic. But again, remember, if people have an MTHFR deficiency, they are not able to get to the bioactive form of vitamin B9. And particularly if they're consuming lots of folic acid, they are going to have an accumulation of folic acid and a deficiency of the necessary 5-methylfolate. So again, we talked about, again, if you are MTHFR deficient, that means that you are going to have decreased levels of 5-methylfolate. And that, again, is going to be problematic because 5-methylfolate is necessary for converting homocysteine into methionine, and we're going to look on the next slide why methionine is important in this SAMe pathway. So again, we learned about this in the context of MTHFR because homocysteine levels, elevated homocysteine levels from MTHFR deficient individuals who are not going to have sufficient levels of 5-methylfolate put you at an increased risk for cardiovascular disease. And not only do we have this little story that we talked about, but 5-methylfolate is really important as a biological methylating agent. So again, we're going to look at this a little bit more detail with this story of methionine, but we're going to see in our next learning unit, we're going to revisit this concept because 5-methylfolate is important for the synthesis of DTTP nucleotide, so the thymine nucleotide. It's also really important, again, for synthesis of certain neurotransmitters and in neural tube formation. So a lot of the things that we talked about last semester, but again, why 5-methylfolate is so important and thus why functional MTHFR is very important. All right, so a little bit more detail as to the methionine story here. So this is going to be sort of a little bit of a cyclic process here. So again, we're going to talk about this cyclic process going in this direction. We're going to talk about the amino acid methionine here, but I'm going to start with this story down here with, again, homocysteine. So homocysteine, again, needs 5-methylfolate in order to provide that methyl group in order to generate methionine. So again, just why is this called homocysteine? We really didn't address this last semester when we learned about MTHFR because we really hadn't done amino acid structures yet. But if you look at what the structure of cysteine is, cysteine has a single methylene group here before we have our thiol functional group. So homocysteine is just a homolog of that, where we have the same functional group at the end, but it's 2 methylene groups that we have here. So this is not cysteine, right? But it's a homolog of cysteine that once it's methylated actually becomes methionine. So again, if you are MTHFR deficient and you have insufficient levels of biologically available 5-methylfolate, you are going to have increased levels of homocysteine and decreased levels of methionine. So why is methionine important? Well, we already know, and we've talked about before, that methionine is sort of the initial starting amino acid for lots of protein synthesis. But it's also really important here because methionine is important to make S-adenosyl methionine, or what we call SAM. SAM is a biological methylating agent. So even though you can think about methionine as having that methyl group that it could use to transfer, it's not going to do it in its amino acid form. You're actually going to basically take an adenine residue and then you're going to attach a methionine at carbon 6. You don't need to know those details, but this is really those two pieces together, and when they come together and you create S-adenosyl methionine, that is what we need to do a lot of this important biological methylating. So kind of completing this circle here, after you do your biological methylation, and there's about 40 known methylating reactions that require this SAM, which is also sometimes called SAMe, then you generate S-adenosyl homocysteine. So again, you get back to having this free thiol group, and then hydrolysis is going to actually remove that adenine piece, and you get to homocysteine here. So again, we've got this nice cycle that we have here that unfortunately can be negatively impacted by individuals who are MTHFR deficient. All right, so another story to kind of visit here is this story of people who have PKU. So PKU stands for phenylketonuria, which means you basically have increased levels of phenylalanine in your blood. Okay, about 1 in 12,000 infants are actually born with this. This is one of the tests that they do when they test for sort of inborn errors of metabolism. When they do a heel stick test on an infant, they're looking for diseases, metabolic disorders, and this happens to be one of them. So individuals who have PKU are deficient in this enzyme called PAH, which is phenylalanine hydroxylase. I do want you to remember the name of that enzyme. So people who have PKU are deficient in PAH. Remember, anytime we have an enzyme deficiency, that means we are going to build up the precursor, which in this case is phenylalanine, and we are going to decrease the concentration of product, which in this case is tyrosine. So even though tyrosine is not considered to be an essential amino acid because we can make it from phenylalanine, individuals who have PKU will have decreased levels of tyrosine. So let's take a second then and talk about why individuals have certain symptoms with PKU. Some of them are related to increased levels of phenylalanine. Some of them are related to decreased levels of tyrosine. So again, a lot of times when you have elevated levels of something in your blood, your body tries to find ways to get rid of it. So one of the things that your body can do if you have excess levels of phenylalanine is it converts it to this phenylacetic acid. And your body tries to sort of excrete this, and this can actually result in a musty smell, not only in urine, but in sweat and so forth, as your body tries to rid itself of this excess metabolite. Something else to think about is phenylalanine is what we call an LNAA, a large neutral amino acid. There's lots of LNAA transporters in your brain because lots of neurotransmitters are actually derived from both phenylalanine and tyrosine. And so when you have excess levels of phenylalanine, they are going to preferentially saturate these LNAA transporters in your brain, and we're not going to have any uptake or limited uptake of other large neutral amino acids, and that has a negative impact on neurotransmitter synthesis. So again, we're going to see on the flip side over here why that is problematic when we look at tyrosine here. So again, decreased levels of tyrosine. Tyrosine is necessary for the production of melanin. So people who have PKU tend to have what we call hypopigmentation. Because they don't produce sufficient levels of melanin, they tend to have very, very light colored skin. In addition, when they don't have sufficient tyrosine, tyrosine is used to synthesize both dopamine and norepinephrine. So again, we're going to have insufficient levels of these neurotransmitters or hormones, and so that's going to be problematic from sort of a neurological standpoint. All right, so a couple of things to think about from a dietary standpoint. People who have PKU have to avoid anything with aspartame in it. Remember in our last learning unit, I think we looked at the structure, or we revisited the structure of aspartame. Aspartame, right, is just phenylalanine with aspartic acid, and that aspartic acid, again, is made into that methyl ester. But the important piece to this story is more that we've got, when we hydrolyze that peptide linkage and that little dipeptide in that artificial sweetener, we generate high levels or unusually high levels of phenylalanine compared to what's found naturally. And people who have PKU have a hard time, again, metabolizing that. So you'll notice anything that contains aspartame, and remember aspartame is the sort of chemical name for the trade-named NutraSweet. So anything that was made with NutraSweet or has aspartame in it, there'll be a warning label on there saying, warning phenylketonuria, this product contains phenylalanine. From a dietary standpoint, not only do you have to avoid NutraSweet or aspartame, but you'll tend to increase your diet with fruits and vegetables, which tend to be not as rich in phenylalanine as, say, meat protein sources are. And oftentimes you'll see supplementation with tyrosine to kind of get around the fact that you've got an inability to naturally produce tyrosine. All right. So how do we actually make some amino acids? So to highlight here, of the 20 amino acids, and you don't need to know which ones are which, but I do want you to know that it's pretty much an even split, 10 and 10, between amino acids that are not essential, meaning we can make them ourselves, and amino acids that are essential, meaning we have to consume them from our diet. And hopefully you'll recognize that some of these are obviously non-essential, things like alanine and aspartate and glutamate, because we know what their amino acid kind of counterparts are, or their alpha keto acid counterparts are, right? So we clearly know we've got pyruvate that we can get from glycolysis. We've got oxaloacetate and alpha ketoglutarate that we can get from the citric acid cycle. So some of these should make sense based on things that we've talked about. But let's talk about how we would make amino acids. And again, we're going to use these transaminases again. And what we're going to see here is an important function for glutamate or glutamic acid. Again, we're going to use glutamic acid as a nitrogen source, and as long as we have the alpha keto acid to make any given amino acid, we can do this transaminase chemistry, freeing up alpha ketoglutarate there, and then taking the nitrogen from this glutamic acid here, putting it with that alpha keto acid to generate our new amino acid. So that's how we can synthesize amino acids. Okay, so that's really the recipe for an amino acid, is we can take the alpha keto acid skeleton, that existing skeleton, and then we take the nitrogen from an existing amino acid, and we specifically take it from glutamate. Okay, so why this is interesting and why we're talking about it is, you guys know that there are 4 taste receptors that you learned about probably in elementary school. There's salt and sweet, which we actually know we have because they often allow us to tap into both macronutrients, that is why we have sweet receptors, and then micronutrients for salts. Those are usually important things that we want to consume in our diet. So we've got taste receptors for those. And then we've got sour and bitter. Those tend to be taste receptors for things we want to avoid. A lot of alkaloids, which are poisons and toxins in nature, are going to signal those taste receptors. So again, those taste receptors are really important for signaling things that we want to have and things that we don't want to have. So one of the things that was recently identified is there's a 5th taste receptor, and it's the umami receptor. And that really is a taste for glutamate. And so monosodium glutamate, which you've probably heard of as a flavor enhancer, is actually glutamic acid in its deprotonated form. So again, if we look at what the charges would be on this, the net would be -1. So that's why there's 1 sodium. So monosodium glutamate. So that's why this is a flavor enhancer, because you actually have a taste receptor for that molecule. So again, here's just a couple little stories. Sometimes people have problems with MSG. And too much MSG can again be problematic. And why that can be problematic is you can have an excess of neurotransmitter synthesis. This can cause an overstimulation of neurons. And so some people who have issues with MSG will report headaches. They'll report sensitivity to heat. They'll have like flushing that they'll have on their face, all things that can be related to sort of these overstimulated neurons. And there's actually a good bit of research that links MSG with chronic pain. So the last little story to sort of talk about here is how you actually might get MSG. Like you can actually walk in and on the shelf just buy MSG as a flavor enhancer. And so often how it's made is by degradation of proteins and then sort of isolating just the MSG from it. But one of the things that's interesting to think about is if the protein source is something that's non-animal, so for example, yeast, soy, or vegetable, even though you do acid hydrolysis and you isolate glutamic acid, you are often isolating the D stereoisomer over it. Remember, we have D sugars in our body and L amino acids. So this is not a bioavailable form, which could potentially be another reason why some people have problems with it. So if you wanted to consume MSG, it's best to get it from a natural source. So if anyone is able to obtain it from enzyme sort of degraded collagen, that is going to give us the L-glutamic acid, which is going to be much more bioavailable and less kind of stressing on the body than having sort of the D flavor of that. All right, so a few other little stories here just to kind of wrap up our discussion on proteins here. Let's talk about heme. And when I say a recipe for heme, I don't want you to be able to think about or repeat anything that we see here, but I do want you to know that the sources of our carbon atoms and our nitrogen atoms are familiar pieces. We make heme using carbon atoms from succinyl-CoA and then nitrogen atoms from glycine. So again, I'm not going to go through any of the detail of this, but all of this is enzymatically zipped together to make heme using, again, carbon atoms from a citric acid cycle intermediate and then nitrogen atoms from the simplest amino acid from nitrogen or from glycine. So just a little kind of recap of, well, why does your body have heme? 2 stories here, and then I'll even just add one other one that we didn't mention in class. Obviously, your red blood cells need heme because that is the oxygen carrying species. That is the ligand to bind oxygen with that iron in hemoglobin. In liver, we talked about the detox enzymes that we have in that class of enzymes called cytochromes P450. So that's really important. And beyond specific tissues here or cell types, remember that we've got cytochrome C that is in our mitochondria and the electron transport chain. So that is another sort of necessary place where we see heme in some biochemistry there. So we did spend a little bit more time sort of talking about heme degradation. So, why might you be, discarding or getting rid of heme? Well, red blood cells only have about a 60 to 90 day lifetime. So once those are turned over, heme needs to be degraded. In addition, if you have any bruising or cellular or tissue damage, you actually are going to have some heme that you need to get rid of there. And so heme, remember, is going to be very red in color. So when you first have a bruise, you're actually going to see that red color. And as that bruise heals, you're going to see it undergo some color changes. And we're going to see the color changes that we actually have highlighted here. Again, bruises are going to start out kind of reddish purplish, then they'll eventually become green and yellow. And what you're actually seeing within the trapped heme that we have within this damaged tissue is you're seeing sort of the breakdown of that heme product into ultimately bilirubin, what we have here. So what I've highlighted are sort of names that I do want you to know. I do want you to know, obviously, that we start with heme and that heme is red in color. But part of the degradation process generates a green product that we call biliverdin, which I don't need you to know that name, but I do want you to know the yellow product that we make from heme degradation, and that's called bilirubin. And you may have heard of bilirubin because excess levels of bilirubin lead to what we call jaundice. So normally what will happen is bilirubin is going to be actually excreted in both urine and feces, and so your body can get rid of it. But this is a process that requires, a well-functioning liver. And so again, whether you're maybe an infant that is not producing sufficient urine, or you're somebody who has liver dysfunction, jaundice happens when you have a deposition of excess bilirubin in your skin, and you really can see it in the whites of your eyes and so forth. So again, jaundice is a buildup of excess bilirubin in skin tissues because you're not able to excrete it. But kind of looking a little bit further at sort of the normal excretion product, you basically can have some heme degradation products are going to make their way into waste product in urine through your kidney. And again, some of this is going to be responsible for the yellow color of your urine. And then some of it will find its way into your gastrointestinal tract and will actually be responsible for the brown color, or some of the brown color that we see in stool. All right, just a few other little sort of concepts that we talked about, kind of very big picture here at the end, is we're basically going to have 5 important atoms that we're going to see here that are responsible for most of our macros. We really haven't talked about phosphorus too much yet. Our last learning unit here for our next exam is going to be on nucleic acids. But again, when we think about carbohydrates, we're going to have carbon, hydrogen, oxygen. We're basically going to have lipids having just carbon and hydrogen. Once we get to proteins, we're going to introduce nitrogen. And then when we get to nucleic acids, we're going to have phosphorus there as well. Obviously, sulfur is an important atom here. But for the little story I'm going to tell here, I'm just going to focus on these 5 atoms that we find in our macros. So I want to talk about for a second where we get these kind of from our environment. And at the end of the day, we consume things, right? We need to consume living things to largely provide these. But at the end of the day, the main source of carbon in the biosphere is going to be from carbon dioxide. Nitrogen is going to be from elemental nitrogen. And we've got lots of water and oxygen that are going to provide hydrogen and oxygen. And then inorganic phosphate can be a source of phosphorus. So I'm just kind of highlighting that 4 out of the 5 of these are gases that are present in the air. But this can be problematic because specifically 2 of them are fairly unreactive. Carbon dioxide is nonpolar and fairly unreactive, and nitrogen is one of the most unreactive gases, right? We know this. If you do any organic synthesis and you want to put it in an inert atmosphere, you do your chemistry under nitrogen. So sort of some big questions and kind of pulling some stories together of things we've already talked about and introducing a new one is two really, really important enzymes in the sort of biological world are the enzymes responsible for fixing nitrogen and the enzyme responsible for fixing carbon dioxide. So let's just kind of come down here first because we've already learned about this enzyme, right? We learned about in the Calvin cycle, Rubisco is the rate controlling step, and that's also the step responsible for fixing that fairly unreactive and unavailable carbon dioxide into carbohydrates, which we can then use as fuel. Okay, again, something else just to highlight is even though we don't have the ability to take carbon dioxide and use it directly and make it into carbohydrates, we do have the ability via carbonic anhydrase to convert carbon dioxide into bicarbonate, and we can use that as a single carbon source. We've seen that in a lot of different processes where we can use bicarbonate to build things, but we're not going to be able to build it into carbohydrates to use as fuel. So it's really important, again, plants have the ability to do that carbon fixation chemistry with Rubisco. So the last little story here dealing with nitrogen is the enzyme nitrogenase. So as much as Rubisco is the money-making enzyme for carbon fixation, nitrogenase is the money-making enzyme for nitrogen fixation. Again, taking this fairly unreactive nitrogen gas and making it into the highly bioavailable ammonia. Now, again, this is not something even plants have the ability to do themselves. Usually they will have sort of a commensal nitrogen-fixing bacteria that might live in their root nodules. Otherwise, you probably have to provide a nitrogen source to plants as fertilizer. So again, really important enzyme that's part of what we call this nitrogen cycle here. Okay. So last couple little stories we mentioned just briefly in a homework question here talking about nitric oxide. Nitric oxide is a really important biological molecule in your body because it is a potent free radical. So if you draw the Lewis structure for this, you'll see that there's an unpaired electron there. It's actually made from the amino acid arginine. And so this is a really important molecule that has several important functions. It's an important vasodilator, so that is how Viagra is able to actually do its job, as it sort of taps into this ability to increase the synthesis of nitric oxide. It's a really important sort of signaling molecule in your central nervous system, and it's actually a really important component of your immune system, as it's a very potent cytotoxin. So just kind of another nitrogen story that we have here. And the last story that we sort of talked about was, again, another amino acid sort of derivative. And again, this is not a naturally derived derivative. Glyphosate is really what we think about as a methyl phosphate of glycine. So what I've got boxed in blue here is really what the amino acid glycine would be. And if we added a methyl group and then a phosphate, we have what's called glyphosate. So glyphosate, for a long time, people kind of were calling glyphosate this 1 in 100-year discovery. They were saying glyphosate is to agriculture what penicillin was to medicine. It was this massive sort of discovery. What glyphosate really is, it's a potent broad-spectrum herbicide and a desiccant. So initially, again, it was used very heavily as an herbicide because it will kill plants. And again, if you don't want them to kill your crops, if you use genetically modified organisms that are resistant to glyphosate, then you can successfully grow your crops. You can throw a whole bunch of glyphosate on it. It'll kill the things that you don't want to be there. And voila, you've just got, an amazing, yield of the crop that you want. Of interest here, how this kills plants is it interferes with the synthesis of these hydrophobic amino acids, and so that prevents germination of seeds and no plant growth. But again, if you are a GMO plant and you are glyphosate resistant, then you can grow just fine. And so kind of the conflict of interest that we have here is the company that was responsible for making the plant that was resistant to glyphosate was also responsible for creating the herbicide that you'd use, so making the glyphosate. Both of them, again, being patented, and again, farmers having to kind of invest in both of these species. So huge conflict of interest. We talked about the documentary Food Inc. as sort of a great story about glyphosate. Mentioning a little bit about the desiccant properties. Again, a desiccant is something that dries things out. So this is sort of a not initially intended use of glyphosate, but again, it's known to dry things out. So even though we don't need to put glyphosate on some crops for sort of the herbicide functions, it's used very heavily on sugar beets because it dries them out, which allows them to increase the sugar concentration. And it's also used to dry grain crops. And again, the thought is we dry these out in the field so we don't have to worry about drying them indoors. We don't have mold issues, things like that. So it was really thought to be sort of this windfall in terms of a discovery that just had so many important aspects to it. But research started accumulating that this molecule was probably problematic, and the World Health Organization actually in 2015 identified it as a probable carcinogen in humans. And so there's been a lot that has kind of come to play since that. One of the take-home questions I think you guys are going to have as part of your take-home has to deal with this glyphosate story. And so one of the things to think about is it is an incredibly pervasive and persistent sort of toxin really in our environment. It is carried by the wind. It's found on every single continent. And it's really, really something. It doesn't break down in the environment. So it's really kind of a sad story to think about. All right, so that wraps up our story on proteins and amino acid metabolism.